Hello everyone, this is Ravindra Babu. Welcome back to my channel Silver 90s by Unit. By the last video, we successfully completed the discussion about DNA and RNA. Now let us move on to the heart of molecular basis of inheritance, that is a central dogma of molecular biology. The central dogma of molecular biology consists of three major events. First one is a replication, where genetic information gets transferred from a DNA molecule to another DNA molecule. Nothing but a parental DNA molecule gets synthesizes a daughter DNA molecule. And the second event is a transcription, where the genetic information which are present in the DNA gets transcribed to mRNA, nothing but a synthesis of mRNA from a DNA molecule. And the third event is a translation, where genetic information gets transferred from RNA to a protein molecule. Nothing but the genetic information of a nucleotide language gets translated into amino acid language. This is what a central dogma of molecular biology you know very well. Now let us start the first event of central dogma of molecular biology, that is a DNA replication or replication of DNA. The DNA replication means simply it is a synthesis of DNA. The synthesis or replication of DNA takes place in S phase, means synthesis phase of interphase of cell cycle. Here, let us discuss this DNA replication in brief by observing the interphase of a mitotic cell division. Here, I am considering this as the cell of human being, which is going to involve in a mitotic division. Here, before going to involve in mitotic division, first it has to enter into interphase. Here, this consists of 46 chromosomes in 23 pairs. Here, I am taking only one chromosome. That one chromosome is having two chromatids, right? This is one chromatid and this is the another chromatid. This type of chromosomes are called 2C chromosomes, and the cell in this condition is called 2C condition. These 2C chromosomes during this S phase of interphase of cell cycle undergoes a replication and become 4C chromosomes like this, nothing but a chromosome having 4 chromatids. So this is a cell having 4C chromosomes after interphase. Nothing but what? Here the 46 chromosomes of this cell are in 2C condition. After the interphase, especially after S phase, the 46 chromosomes become a 4C chromosomes. This is what actually the synthesis of DNA, where the DNA only will become double, the number of chromatids only will become double, but not the chromosomes. Remember this point. And in the year 1953 itself, Watson and Crick proposed this a scheme of a DNA replication, which is a semi-conservative model of DNA replication, while proposing the 3D model of a DNA. According to them, the DNA mode of replication is a semi-conservative model of DNA replication. According to the semi-conservative model of replication, the two strands of DNA before undergoing replication gets separated each other. So this is a double-stranded DNA molecule, and this is the sequence of nucleotides of both strands. First let us take sequence of nucleotides of this strand. So this is having A, T, G, C, T sequence of nucleotides, and the sequence of nucleotides of opposite strands are what? T, A, C, G, A. As both of the strands are complementary to each other, they follow a rule of base pairing and complementarity. so that if you know the sequence of one strand of DNA, automatically you will come to know the sequence of nucleotides of opposite strand of DNA molecule. Now this DNA molecule is going to involve in replication. During replication, both the strands of this parental DNA gets separated like this. This is one strand and this is the another strand. What is the sequence of nucleotides here? Here in this strand TA, CG, A, and the sequence of nucleotides of this strand as AT, GC, T here. These two separated DNA strands acts as a templates. Template means a parental DNA strand on which a new strand gets synthesized. These are the two parental strands acting as a templates here. Say, if it acts as a template, it synthesizes a new DNA strand. The new DNA strand will have a complementary nucleotides to this parental DNA strand. So here the T, here A will come, T, G, C and T. Likewise, these parental strands also synthesize a new DNA strand, complementary DNA strand, which will have T, A, C, G and A. If you carefully observe these two daughter DNA molecules coming from this parental DNA, here both are having same sequence of base pairs. TAAT, CG, GC, AT, TAAT, CG, GC, AT, and both are 100% completely identical to each other, and at the same time both of these daughter DNA molecules are identical to the parental DNA, so that the synthesis of DNA is called as replication. Replication. And one more point you can observe here, that is the two daughter DNA molecules, whatever produced from this parental DNA molecule, are conserving one parental strand and having a newly synthesized DNA strand. Here also, this DNA molecule also conserving one parental strand as such and synthesizing a new DNA strand. Half of the DNA is a parental and half of the DNA is a newly synthesized in every daughter DNA molecule, so that we can call this replication as semi-conservative model of replication. Semi-conservative model of replication. Along with this model of DNA replication proposed by Watson and Crick in the year 1953, there were some scientists who proposed different methods of DNA replications, and the second one is conservative model of DNA replication. According to this conservative model of DNA replication, the two daughter DNA molecules whichever produced from the parental DNA, one conserves a parental strands as such and one consists a completely newly synthesized strands like this. So this is a double stranded DNA molecule having AT, G sequences and here T, A, C sequences. If it undergoes a replication, one daughter DNA molecule will be produced which conserve completely parental strands as such, and another DNA molecule will be of newly synthesized like this. This is what we call a conservative model of DNA replication. This was proposed before the semi-conservative model of replication, and there is one more model of DNA replication, that is a dispersive model of DNA replication. This dispersive model of DNA replication says that the two daughter DNA molecules whichever produced from a parental DNA molecule will have the segments or patches of a parental DNA and a newly synthesized DNA. like this. If it undergoes replication, say daughter DNA molecules will be formed having patches of parental strands and newly synthesized DNA strands like this. Okay, these are the segments of DNA coming from parental DNA, and these are the segments of DNA which are being synthesized newly. Among these three models of DNA replication, the two models, conservative model and a dispersive model of replications, could not able to give experimental evidence, but semi-conservative model of DNA replication was experimentally proved by Michelson and Stahl later. The experimental proof for the semi-conservative model of DNA replication will be discussed in the next video. Thanks for watching this video. Please like, share and subscribe my channel.